Good afternoon, everybody. First of all, I would like to thank organizers for giving me this opportunity to share with you some results, what we are doing in Wroclaw University of Environmental and Life Sciences. Right now, I will skip a little bit from enzymes to whole cells, of microorganisms, especially to fungi. Yes, so from the morning, my colleagues Jarek, Agata, Kinga were talking about some protein engineering, biological, molecular biology, but I will share with you some results with whole cells. Okay. Yes, so as all of you know, response we can obtain via chemical synthesis or biocatalysis. Of course, this is we know about this. As recently, from last decade, some biotechnology methods receive more and more attention. So I would like to focus today on fungi. Yes, on fungi. So we are thinking that these biocatalysts are really efficient, especially because of wide range of enzymes. They can produce pigments and flavors, because I will focus today on talking on this kind of two group of compounds, let's say. We see that fungi, because of their tolerance, flexibility, cheap requirements for their growth, they are really useful and efficient biocatalysts. Okay, so let's start with pigments. Natural pigments consist of wide array of different pigments. Yes, so we have carotenoids, azophyllones, melanins, and others. So, but there is still a problem which basic science and industry haven't overcome to receive a solution of high cost of pigments and low availability of them. So we are trying to use fungi, especially endophytic fungi, which I will tell you in 3 minutes, to use them to obtain different kind of pigments. One project at our department, at our group, is conducting by Dr. El Sayed Ali from Egypt. He received a project. He's with us for 2 years. And so the idea is to obtain from endophytes different kind of pigments. Today, I will talk mainly about Monascus. So Monascus is, of course, known fungi. Monascus rubber is producing edible pigments. We isolated this strain from the plant, and we determined morphologically and then genetically determined this strain. So our results confirmed, of course, that this strain is producing 5 pigments from yellow, orange to red colors. They are these pigments are of azophyllones structure. I will tell you that these pigments are really expensive. So we bought commercially. They are commercially available. We bought this pattern, so we could confirm that they are produced. So our aim was to increase the yield of these pigments. Yes, because industry is really interesting to receive some pigments in really high amounts. So we try to screen how different byproducts, waste, so side streams of agri-industry, agri-food industry, if they are good or which is better. Yeah. So from our results, we saw that the best were potato peel, which were moistened by mineral salt broth. Okay, so after optimization studies, we were focusing on temperature, of incubation period, pH, inoculum concentration, substrate weight, moisture level. We try to increase the yield to find the optimal conditions for producing these pigments. What we did also, because Dr. Said Ali, he's working in Atomic Energy Authority in Cairo, in Egypt. So he's making mutagenesis via gamma irradiation. So after exposition to different doses of gamma irradiation, we obtain some mutants with the increased red pigment yield. So enhancing by really good results that this kind of mutation is providing us a better result, we focused on making mutations in this 1 kilogram dose of gamma irradiation. We received many mutants, and we observed that some of them are producing more red pigment. Especially, I would like to mention about one mutant, M22, which, as we see, is really promising according to red pigment production. Okay, so we compare the parent and the mutant. We saw some kind of differences in the growth, in the spores, in the color, of course. The parent was light red pigmentation, and the mutant was more deeper red pigmentation. Okay, so we try to focus on immobilization of this mutant. So we were looking for different entrapment carriers in submerged fermentation. We check different ways, let's say, or carriers, and the best one was calcium alginate. Okay, then we were trying to focus on different kind of media. So from these experiments, we saw that the best was modified lens medium. Okay, and we checked the number of cycles of fermentation. So it was still we could 8 times grow it and receive red pigments. Never mind if we were using spores of mycelium. Okay, so just recently, in cooperation with Egyptian institution, we are doing some kind of nanoconjugates. So we are trying to make some kind of nanoconjugates with the red pigment and zinc, copper, or magnesium sulfate with gamma irradiation, and then check antimicrobial activity of these conjugates. Okay, so my title was Pigments and Flavors. Of course, we can talk about pigments, and we need to take 1 or 2 pigments and talk about this, but I would like to give you an overview what we are doing. So right now I will skip a little bit, but really slowly. So I was talking about Monascus rubber. So if you are interested in this strain, but not producing pigments, but producing sesamol, I invite you to go and meet Ewa Szczepańska. She's today with us. She will have a poster today. So it is about biotransformation with whole cells, where we are using piperonilic acid, and we can obtain sesamol. So this compound sesamol is right now, you can get it from sesame, from the sesame oil. And this is a compound with great biological activity. So we made a patent application, and but if you are interested, please visit Ewa. Okay, so let's skip and let's go to flavor and fragrance compounds. So I would like to tell you today about producing of this propenyl benzene from propenyl benzene compounds, the aldehydes. So the really known aldehydes and really interesting from the industrial point of view are vanillin, piperonal, but also others. They are, of course, we know how they smell, and they are really with pleasant flavors. So we could think that the common substrates could be propenyl benzenes for this biotransformation. So to obtain corresponding aldehydes, we could start with isosafrole, anethone, and isoeugenol or others. But these compounds has antimicrobial properties. So the fungi, or generally saying microorganisms, they don't want to transform these compounds. So, but we tried. Yes, we tried. We check plenty of biotransformation, and only isosafrole could be transformed with Trametes hirsuta to piperonal, and we also obtain some dihydroxy derivatives, so diol, let's say shortly. So from the point of view of biotransformation, so the yields with 50 or 60 isolation yields, 50, 62% are really good. So we were happy with these results, but we wanted to make some general route for obtaining these aldehydes. So we saw, as I mentioned, that we obtain also diol. So we decided, okay, so let's make diols with different substitutions, yes, with different compounds, and then hopefully it will be less toxic. So we will check if they can be transformed to corresponding aldehydes. But no, the main product was hydroxyketone, as you can, as you can see. Okay, so we were checking bacteria and fungi. So if you are interested in this two-step synthesis, we made it with collaboration of a group from Politecnico di Milano. So it was starting from propenyl benzenes, we obtained diols, and then wholesale biotransformation to hydroxyketone. We obtained several kind of compounds, and then we checked their biological activity, different biological activity. So if you are interested, please visit this paper. Okay, so we couldn't obtain aldehydes, yes, but we need to develop another route. So we decided, okay, so let's start from carboxylic acid. So let's focus on car activity. Yeah, so we took the acids, yes, and we use whole cells of biotransformation using Basidiomycota strain. So this was a good idea because, as you see, we obtained different kind of aldehydes. Okay, so we use 5 substrates and then 12 microorganisms, and from these results, we obtained these aldehydes. But only to show you that during the biotransformation, there were also formed other products. So of course, the reduction was going farther from aldehyde to alcohol, but also other products in some cases. So this is only to show you that we need to monitor the biotransformation and stop the reaction in appropriate moment to have these aldehydes. Okay, so finally we received different kind of aldehydes with the yields, satisfactory yields. So the best biocatalysts were Pleurotus, Pycnoporus, Lepista nuda, and our findings show that different strains belonging to the same fungal species can perform the investigated biotransformation in different manner. Yeah, so it is crucial to focus on really strains. Yeah, so this is the let's say important remark. Okay, so another topic also. Eva is involved in this project. She's a PI of this project. So we want to obtain flavor fragrance compounds. So how to obtain vanillin? So she receives money from the institution, yes, and CBR, that her goal is to obtain vanillin from different byproducts, agro-food industry byproducts. So she developed different byproducts. In these byproducts, we have lignins, then ferulic acid. So later we want to obtain vanillin or vanillic acid, but it depends. So the project still lasts. It has 5 phases, but if you are interested, please talk with Eva during the poster session or coffee breaks. Okay, the other project which already was submitted to Opus, so National Center institution funding this project, it is connected also with endophytes, but producing flavors, not pigments. So we see real potential in these endophytes. So ending, I would like to thank my colleagues from Italy, from Egypt, and of course my Polish team for this collaborating research. And finally, I would like to thank you all for your kind attention. Thank you.